In today's video, we will be exploring our Omics AI Assistant. This application brings the talk-to-your-data workflow to life with its intelligent agent-based orchestration, allowing users to analyze, visualize, and interact with multi-omics data in a very fluid way, much different from the dashboard-based approach they typically used in the past. Let's start by asking where we should start. The chatbot responds with a list of all the relevant available agents, along with sample prompts to interact with each of them. Every prompt to this assistant goes through a flow orchestrator agent to determine which agent is best equipped to handle the user's prompt. We'll begin our exploration by asking the chatbot to describe a gene expression profiling dataset that the user has uploaded, and suggest some visualizations for it. As you can see, this prompt gets routed to the data visualization agent. The agent evaluates the file and infers that it is a dataset containing gene expression levels for different probes, and suggests a few useful visualizations. Based on the suggestions, let's choose the first one, a heat map. We provide some instructions about how we would like to see the heat map visualized. Because Dial integrates with custom visualization renders like the Plotly Python graphing library, we can generate code to invoke its heat map generation function. As you can see, it generates the heat map and returns the Plotly chart. As humans, we can get a good qualitative feel for the heat map just by looking at it. To get more quantitative with this analysis, we can ask the assistant to interpret it, including a specific question about the gene expression levels. This calls our interpretation agent, which in turn provides some observations and answers our prompt. Moving on, let's ask it specific questions about our dataset. We ask how many unique genes and probes are in there, the mean expression level, the genes with the highest standard deviation, and some descriptive statistics for all of them. This prompt is routed to the data analytics agent, which provides a detailed explanation of its methodology, actually a plan to answer all the questions, and shows you all the code that is generated in order to do just that. As a result, we see the genes with the largest standard deviations of expression levels. Such genes can be some of the most interesting for research, so let's focus on these 10. Let's take a look at the cognate proteins for these genes. We retrieve the UniProt IDs for these proteins using our Gene IDs mapping agent, returning them in JSON format using the UniProt API. Let's pick a protein from the list. Our Go annotations agent consults the gene ontology for information regarding molecular function, biological process, and intracellular location pertinent to that protein, and retrieves it. We show the resulting graphic directly in Dial, and we can also ask the assistant to interpret it for us. While this Go annotation visualization is pretty neat, it's generated by an external API. Let's take a look at some other visualizations that our assistants actually build on demand for us. We'll start with our Reactome biomedical pathways visualization. Here, we ask our Reactome visualization agent to query the reactome.org API to provide pathways in which each of the 10 genes mentioned previously participates. Next, the agent extracts that data into a table and graph, and if any two genes both participate in the shared pathway, an edge is created between the nodes to show that relationship explicitly. We use Plotly again to render this network graph. As we can see, even among these 10 genes, the gene CDK1 is a hub. It participates in a lot of pathways with the other genes. Let's confirm this by building a bar chart of the number of pathways each gene contains. We can now see that CDK1 participates in 218 distinct biomedical pathways. Finally, let's ask our AlphaFold visualization agent to show the predicted structure of this protein. We can see that it gets the gene-to-protein mapping from the previous mapping graph. Then, we use the UniProt database to get the structure as predicted by the protein databank, and visualize the structure in 3D using custom code and Plotly libraries. Thank you.